Okay. We are live on the air. Next week, when you come into the lab, and this is next week, what beverage are you bringing in? Sorry? Someone left us behind. One of these things, isn't it? Now, I'd strongly encourage you to bring something in that has some kind of colour, red, blue, whatever, green, preferably not colourless, but be careful with the nutrition label. Remember, we need one of those. And this time round, you need to not only note the colour, because in the lab next week, you'll be doing a spectrum, taking the spectrum of your red dye and your beverage. You're looking for interference. But I'd be awfully careful with these Gatorade-y things, because what other things do they throw in there? Sorry? Some, yes. Yeah. What else? If you're sweating a lot, what are they trying to replace? Salts. Do you think that's likely to affect your assay? I'd be very cautious. You might want to do dilutions yet again. Keep that in mind when you get into the lab next week. Now, next week, the team should be mixed up a little bit as well. Next week is pretty straightforward, but you will have to do the spectrum, so keep that in mind. You need to do a spectrum on that, and the team leaders will need to talk about that in their particular outcomes. Now, we have a slightly busy day today, so I'm not going to waste too much more time. We're going to get on with fatty acids. Last week, we talked about the notion that fats are ingested, digested, sent through the system. Is that a bit better now, this lapel thing? Not too quiet for you up the back? Microphone's all right? Digested, cut up, sent in, repackaged, put back together, sent out, run around, doing various things, and how you make fats. Well, today we're going to see how you modify them and break them down and send them out into the circulation. So we're basically in this part of the pathway, and I keep talking about this acetyl-CoA, kind of pivotal in there. What's that? 2 carbons. I can go into the electron transport chain via the citric acid cycle, or I could make ketone bodies. We have to deal with processing these as well, particularly in cells. Let's start with some modifications. Now, I'm focused mostly on humans. One, because bugs do different things, so do plants, ruminants, i.e. your cows, your sheep, goats, camels, pigs and the like, and horses. But you need to modify them because your lipid bilayer needs a double bond in it, in that triacylglycerol, to make it more fluid, more mobile. You know you can make a 16 carbon palmitate, but it might need to be elongated in some parts of the body, and you note down the bottom, your brain has to make some slightly elongated ones, adding a few more carbons, and your brain does that specifically for insulation purposes. And just jot down in Schwann cells particularly, those ones that wrap around neurons that help with the action potential. We talk a bit about that in detail next semester, and I believe physiology does some of that too. If we look over at the diagram, we can see palmitate you synthesised last week, whereabouts? I'm still home alone, so I don't have to rush home in a hurry. Where do you synthesise your palmitate? In the liver, in the hepatocyte. That will be an exam question now. I know you're not thinking too much. You can do things to it, like remove some double bonds called desaturation. But you're limited in your cells. You can really only desaturate, that is, remove a couple of those hydrogens, usually up to a position 9, 10, that's it. You don't have the enzymes to do anything beyond that. And you mostly do that. That is, you make a palmitate. What does 16 double dot 1 mean, delta 9? 1 double bond, position 9 carbon, isn't it? That'll be an exam question. Some individuals, like cows, can start making things like stearate. Steers, give you an idea. A couple more carbons, there's 2 of them going in there as well. If If you desaturate that, you end up with an 18-1 delta 9. If you elongate further, you can have those longer saturated fats. This ones that go down the brain. Carbon 22 to carbon 24. Very specific long chain fats for insulation purposes. Beyond that, you see the plants there, just put a line through there. You have to ingest that material. You can't make it. Plants can. Linolate, they make an 18-2, 9-12 carbon double bond. Some make more complex, 18-3. These are the polyunsaturated fats, things like canola oil. So my colleagues are working on that. I'm helping them with some confocal, some high-end hyperspectral analysis. They're looking at canola because canola, like cabbage, like cauliflower, like broccoli, has fluorescent molecules in it. It deals with UV light, but it also affects things like your bone density. It affects things like estrogen production. Lots of luciferous molecules, like lots of cabbage, can affect your estrogen levels in males and females. That might be useful for you in that report part A that you might be dealing with now. What molecules there? And we're going to see report part B, other molecules that affect your metabolism. Some desaturation go on to make some of these characters, particularly this thing, arachidonate. You'll see it starts life as this complex one, the eicosanoate, at 20.3. Remember I showed you that horseshoe-shaped fat last week? Yep, they're eicosanoids. I just mentioned one last week, arachidonate. What was unusual about them? You only synthesize them when you need them. You don't store them. But you need some of these molecules supplied to you to actually manufacture them. These are important components for you. I've got some key points for you. You make 16 carbon fats. You sent them out last week with VLDL. What does VLDL stand for? Very low density lipoproteins. Remember that small vehicle, a taxi running around. Palmitate is what you mostly deal with. In this process, you use the palmitate CoA. What's the CoA tell you? It's an activated fat. Now I can do something else with it. If I do something else with it, I can take it to the ER. Which ER am I taking it to? Smooth ER. Rough ER is making polypeptides. You can add a couple more carbons. That's what you've just done over here. You've made stearate, for example. Who was the key regulator in there? Malonyl-CoA. Who was that? 2 carbons coming out. That's the key regulator pathway to make fat for palmitate in liver cells. NADPH, we saw a little bit of that. That proton-electron carrier that helps us as we go through. You done with this one? So yes? Okay. Let's have a look at these, what are termed essential fatty acids. What are they making? Down here, underline the word eicosanoids. These are these extracellular messengers. I showed you one last week. Extracellular messengers that help do things. Prostaglandins affect what you do in some of your hormones, particularly your reproductive hormones. Leukotrienes affect your immune system. Immune cells particularly, like granulocytes, eosinophils. I'll let you do immunology to find out what they do. It also affects things like thromboxanes. That's blood clotting. Again, we talk about that next semester. You synthesize these characters from this base molecule. So see, it says diet. That means you have to ingest it. There's the molecule ingest, and what do you ingest it from? Things like plants and fish, for example. What do you do with it? Well, you take that molecule, you grab some oxygen, stuff you've breathed, some electron carriers, some more protons, you use a desaturase. What are you doing here? You're introducing another double bond. Where do you normally introduce a double bond? Position 9. That's what you can do. You run down another step, you can add a couple of carbons on there, the elongation using malonyl-CoA. Remember that worked for fatty acid synthesis? 3 carbons coming in, rip one off, I can start making a longer chain fat. We run down to this thing, we've made our molecule, and you see all those double bonds there, that's a nice U-turn in the molecule. The U-turn is critical. If it's not a U-turn molecule looking like that, it doesn't work properly. It needs those double bonds there. Excuse me. So these are essential for your diet. You can't synthesize the base molecule, but you modify it to help you do the work you do. Which is why you should have a balanced diet along with lots of plant oils, but not too much plant oil like olive oil and canola oil, because remember, it's full of fat, isn't it? And what do you do with fat? You store a lot of it. We're done with this one? Not quite. Now, we could spend a couple of weeks just talking about these kinds of facts, their activity. For this unit, this is the level I'm asking for. You understand what these eicosanoids do. They're extracellular messengers. They work in different levels, your immune system, your neuronal system, and also blood clotting system. You don't synthesize and store them. You synthesize and use them immediately. You must have the base molecule in your diet. You modify them. Where are you likely to modify them? In the smooth ER. Now ask yourself, if you're a nutritionist or worried about your diet, you weren't having enough plant or fish oils, what's likely to happen? Well, you may not get clotting sufficiently. You might alter your cycle, for example, your estrogen levels. You might alter your immune system, so must have a balanced diet, which is what I'm going to hammer the Hawkesbury students on, because a lot of them are food and nutrition students. Can I move to the next one? Okay. This process of desaturation, let's get back to some grunt biochemistry. Desaturation, what are we doing? Taking off hydrogens, aren't we? Producing double bonds. Let's look at the top image here. This thing is a long chain fat. What you see on the end is the CoA. So what does it tell me about that fat? It's activated. What are you doing with this fat? Well, Lennon just colour-coded the brown bit there. You brought in oxygen and protons. What do we want to do? We want to generate a double bond in that position. Where's it likely to be? Position 9, carbon. How do you do that? Well, you need this fatty acyl CoA desaturase. There's rocket science. Desaturating it, CoA because it's activated, fatty acid component. But how do you go about it? Well, it's complicated. And it requires things that you wouldn't have normally expected. Let's have a look at it. It requires the donation, the process here, of some electrons. Where does it get the electrons from? Fe2+. What have you generated here? Fe3+. What do you think the CYT stands for? What does it stand for? Speak up. Cytochrome. What's a cytochrome? When it's at home. Feed up in front of the fire. It's just a complex proteinous molecule normally that has a colour. And you have lots of them in your mitochondria. So when you look at cells down the microscope, you'll notice that the mitochondria look kind of dark, without any stains, anything else added. Why do they look dark? Because they have lots of cytochromes. Those cytochromes tell you they're busy there. One of those cytochromes you want to jot down on this slide is cytochrome C. It moves electrons from one component, protein 3, to protein 4 in the ETC, electron transport chain. These cytochromes are pivotal to moving electrons around, particularly. Here it is involved with this iron component. Where did it start life? With a different cytochrome, in this case using FAD. We're going to see FAD structure shortly. Where does it get its energy from? Well, from NADPH. What's that? An electron-proton carrier. Now that electron-proton carrier donates here, reacts there. It's like this kind of circuit diagram. What does it do? It donates it to this enzyme. What have you just done? Remove some hydrogens. What have you made? Your product? Desalination plant, aren't you? You've generated a double bond, kicked out 2 protons. The oxygen you brought in, you've made water. So every time you desaturate, you generate water. Every time you do that, you use electron-proton carriers as you go. This is important for you, so I need you to note for the exam, you don't just use oxygen to use in your mitochondria to generate ATP. Oxygen's used here to generate double bonds in fats. Is that kind of clear? It's not just to keep you alive, it's to make these fats. We comfortable with that concept? Can I move on? Okay. Now, I showed this last week. I don't want to spend too long on it because we've got lots to get through today. What's going on in this slide? Look over here at this cell. Here's a typical kind of eukaryotic cell. Let's call it an hepatocyte for now. Nucleus doesn't look too circular. There's lots of ER, doesn't look quite like that. Here's some mitochondria. They're moving around, doing all sorts of weird things. They change shape. This mitochondria in this cell, last week I said no fatty acid oxidation. Down below it says fatty acid oxidation. Where it says no fatty acid oxidation, when does that occur? If you're in a liver cell, when does a liver cell not oxidize fat, break them up to generate energy? When they're making fats, exactly. So right next to that, no fatty acid oxidation while you're making fat. That hepatocyte is busy going through glycolysis, through the citric acid cycle, part of it, the citrate, off to malonyl-CoA, and then making fat. Down below that, you see fatty acid oxidation. In bracket, acyl-CoA production, ketone body synthesis, fatty acid elongation. Why in the black? What are you going to put next to those? You're on a high-fat diet or a no-fat diet? Are you burning energy or are you making fat? Here you are in the fasted state. Fasted. Top one, you're in the fed state. You're making fat. Down below where it says fatty acid oxidation, you're in the fasted state. You're starting to use some of that fat, particularly for more than a couple of days. Now you notice the pink writing is kind of important as well. Here we see a little bit of fatty acid elongation in the mitochondria, not much. Most of it occurs, we said already, in the smooth ER. But this smooth ER, write it down, smooth ER. What are you doing also in there? Phospholipid synthesis. That's why we have this stuff over here. Remember the 3 long carbons stick to the glycerol motor? What do? Take off one of those and put a phosphate group on. Polar, hydrophobic. You've got them in all your membranes. You need to recycle them all the time. Flip A's, flip sides. We talk about that next semester as well. You need to make them all the time. That's one of the dominant activities of cells. A major energy use for your cells all the time is turning over lipids. Think about lipids. They're going to oxidise. You leave them there too long, they oxidise. Constantly refreshing, that's what you're doing. Sterol synthesis, things like steroids and the like, where would you make those? Which gland have we talked about already you're likely to make steroids? Adrenal, but where else? Don't be bashful, sorry. Pituitary, no, that makes peptides, sorry. Gonadal, good job, not so bashful. Your ovaries or your testes will make them, depending on which ones you've got, if you still have them. Don't laugh at that. It's not that funny. Down here in the cytosol, what are you doing here? NADPH. We've got some isoprenoids and some more sterol synthesis. I'm sorry, it's clipped off the bottom there. And a little bit more fatty acid synthesis. What does that mean? Down here, didn't we talk about that last week? Malonyl-CoA in the cytosol via the citrate pathway. What did you do? What enzyme did you do? What was the acronym for that enzyme last week? FAS1, particularly for humans, isn't it? FAS for most species, 1 is yours. Only makes palmitate. The other bit was different now, these peroxisomes. Now those are mostly in adipocytes, some muscle cells. What goes on here? Catabolism of very long-chain fatty acids, some branch-chain fats, some D-amino acids and polyamines. What does that mean? The more complex long-chain carbons peroxisomes deal with. You saw last week the truck backed up to the docking station. It was engulfed, sent into a lysosome, and everything's broken down. That's essentially a peroxisome, breaking everything up, producing carbons ready for production of electrons and protons. Can I move to the next one? That's not a question, I hope. Can I move to the next one? Okay. Up the top, have a look at it. There it is, 2 fingers, just for you. Glycerol, phosphate. There it is, 2 fingers just for you. You notice carbon 2, what does it have in it? The commonly occurring double bond. If it didn't have a double bond, it'd be very solid. Double bond makes it a bit more fluid. There's the 3-carbon glycerol. Just circle those 3 carbons, write down glycerol, because we're going to keep coming back to them. Each one of these long-chain fats you see up here has a specific enzyme to cleave them for recycling purposes. This is introducing you the notion of this one's called phospholipase A1. What does it clip off? Carbon 1 lipids. Now that's going to be important in fat cells because fat cells store this stuff. What are you going to do in a fat cell? Clip off each of those long chain carbons so a muscle cell can use them for energy. You down here for carbon 2, it's phospholipase A2. You might have an exam question like that. What does phospholipase do? It cleaves that carbon, releasing the acid function lipid. Down here, phospholipase C. I want you to put next to that PLC, all caps. And don't bother going to the quiz already. It won't be PLC as the acronym start today. PLC, why is that so important? Well, that's an enzyme we see commonly, I want you to write down commonly, in signal transduction. I'm going to introduce you to a little bit of that today. Signal transduction is taking a signal from outside the cell, usually hormones, telling the cell to do something via receptors. There's a whole lot of signalling pathway steps, cascades. PLC is a common one because it often clips off something off of this lipid structure and starts an event, tells the cell to get busy, particularly in the cytoplasm. In this case, PLC clips off this phosphate, releasing the sugar and phosphate moiety. As soon as that does, somebody gets busy. Down here you see phospholipase D. That one's less common. But what does it do? It releases a sugar moiety for someone else to use, like a cell membrane. So this gives you an idea of what goes on in the lipid bilayer. And what I'd like you to do is draw a bit of a box around those long chain carbons there and note hydrophobic. Here they are, hydrophobic. Here's the other layer, hydrophobic. What do? They come together. And we saw that last week. The lipid layer sticks together, doesn't like water, integral membrane proteins, hydrophobic domains through that, and the phosphate groups hanging off that like water. This is mobile. With that double bond, it's very mobile. The mitochondrial membrane, it's less mobile, keeping protons out of the matrix. Now these have issues because we've seen already the important components like eicosanoids, but these have issues because these lipids, I keep saying, break down. I've already hinted at you that your Schwann cells wrap around and around and around your neuronal cells to increase insulation, just like electrical wiring. Here there's intervals called nodes of Ranvier. Action potential, boom, another one, boom, and I'll talk about how that works in sodium transporters next semester. But the issue here is that those don't last forever, just like that butter in the fridge. It's not going to last forever. You always have to turn them over. You're always constantly refreshing them. Otherwise they oxidise, they go rank on you. And the enzymes that do that work, like these characters, if there's a defect in them, what's the topic of your first topic for report part A? Genetic disorders. If you don't have those enzymes working, the lipids aren't turned over, you have ganglioside issues and you don't have brain function working correctly. Again, you wouldn't be in this room. So you must turn them over for most of your life. And I'm here trying to increase your brain size so that as your brain shrinks as you get older, it doesn't shrink to so little that you're non-functional. So these are all important for you, but turnover is critical. Please note that turnover of lipids is critical. You can't leave them there forever. And they're turned over in a matter of hours and days because it's at 37, high oxygen environment essentially. We done with this one? Okay. And how does this go on within you as an organism now, not just the cells? Well, you turn over fats, and again I want you to write on this slide, you're constantly refreshing fats, even the ones in your fat cells. Don't sit there forever. They are constantly being recycled and refreshed. This process, if you look at the schematic, there's adipose tissue right underneath adipocyte. What does your adipose tissue do? It stores these guys, these triacylglycerols. We'll see what happens to them shortly. It recycles them. Please note here, this is the fasted state. You're going to get an exam question that'll talk about the fed, fasted and stress state. Fed, lots of stuff coming in. Fasted, nothing coming in, I have to get things recycling. Stress state, more acute. I have got to stimulate response and release very quickly. What happens in the fasted state? Well, there's my triacylglycerol. What's your body do the longer it fasts? Yes, I use up some proteins, but I'm exercising so I don't lose all those proteins, all that muscle mass. It starts releasing these, but these things are stuck in a fat cell. We're going to see shortly, they don't just get dumped out like that. That thing has to be cut off there, cut off there, cut off there. Each of those fingers go out of the fat cell and the glycerol moiety goes out of the fat cell. Who uses them, we're going to see next. The long chain fats, just write down there 3 times per molecule. That's a long chain fatty acid. For you, what length is that carbon? 16. Where does it go to? Well, your fat depot is usually fairly close to your muscle depot or your mammary glands, so where the work's required for mammary cells. So when you release the fats, they usually go fairly close by. Hopefully they're used by those muscles, heart or otherwise. They're usually carried by that little protein you're using every week as a standard, which is albumin, thank you. BSAs are cows. Yours is HSA, human serum albumin. Good point, though. It's albumin. It carries a bit of fat from the fat cell to the muscle cell. Because if those characters are loose, what do they want to do? Coalesce. So they form lipid droplets in your blood vasculature. When they get into your muscle cell, they start getting broken up into, and just jot down here before acetyl-CoA, beta oxidation. We're seeing that today. What does beta mean? Remember I said the carbon 1, carbon 2 is a beta? You rip off 2 carbons at a time. We're seeing that today. That produces ATP for work. That muscle can do that work, whatever that work is, usually contraction. There's the glycerol. Remember the 3 knuckles? There they go off. Where do they go to? Well, they go to the liver. They don't need to be carried by anybody. What does the liver do to them? Remember, it's 3 carbons. What can you do with 3 carbons if you have 2 of them? Bring them together and make glucose. That's what your liver does. So that triacylglycerol and that triacylglycerol in a fat cell has the fingers taken off, fingers used by muscle, glycerol gets to the liver, you've turned into sugar, haven't you? Now, when I told you earlier you don't turn fat into sugar, it's not quite accurate, is it? The fats you don't turn into sugar, but the glycerol moiety you do. Does that make sense? Think about the 6 carbon molecule from glucose as we run through the liver. Now, this is kind of important for most of your muscle cells, and it adds to, as I point out down here in italics, your not only skeletal or striated muscle, it's different to your smooth muscle around your blood vessel and around your gut, but also your heart muscle. Your heart muscle uses these fats preferentially, but it also uses things like cholesterol and some ketone bodies when it's really desperate. Now, the red blood cell has to do a bit as well. What's different and unusual about your red blood cells? Remember, it's kind of bicuspid, kind of looks like a little lozenge thing pushed in the middle. Why is it pushed in the middle? Sorry? No nucleus and no mitochondria, because that takes up space, full of hemoglobin. So that means the cell's got to use just glycolysis, that is, no mitochondria to make the energy to stay alive, pump things around, like CO2 in, convert, bicarb out. In tissue, in your lungs, bicarb in, CO2 out. That's what your red blood cells do. So this process is taking us through production, producing energy required, but keep in mind the red blood cell is different. Liver's managing everything as always. Muscle uses some. Red blood cell is different. How long does a red blood cell last? Sorry? 140, 150, yeah, varies a little bit depending on those. So its lifespan is relatively short because it doesn't have all the machinery to stay alive. It's being produced in your bone marrow all the time, which if you do the advanced immunology or advanced cell with me, you'll see why. Can I move to the next one? Okay. Let's have a look at this process of balancing between what you do in different tissues. Now, don't write anything just yet, just look at the schematic. On the left-hand side, you see fat. This is an adipocyte. Adipocyte's going to release fatty acids, fingers off they go. Glycerol, off it goes, into the circulation. In that process too, it gets to the circulation and the liver, the master controller, has to manage what you're doing at that point in time. What you've ingested, what you have available to do the work you're supposed to do at that time. In the blood, there's going to be this balancing act as well, but also other characters like these guys. What's your common glucocorticoid? Cortisol, write it down. The other one is corticosterone. Cortisol, remember, it's a transcriptional regulator. It's going to alter gene expression. Tricky character. Just jot down, it's carried around because it's hydrophobic by a molecule called CBG, or AKA transcortin, corticosteroid binding globulin. Doesn't like water. It gets in, you can see it's affecting expression of genes, but it does it differentially, and that's the trouble with things like cortisol. It's dose and tissue specific on what it does to those tissues. In the liver, for example, you can see glucocorticoids, green arrow tells me what? Increase. It's going to increase, enhance transcription of the gene to make this thing, PEPCK. Does anybody know what PEPCK is? Oh, I do. You need to write it down because it'll bite you. Phosphoenol pyruvate carboxy kinase. You got all that? Good. You're going to be tested on it. Phospho, PH, not F. Phosphoenol, E-N-O-L. Pyruvate, kind of rings a bell now, doesn't it? Carboxy, lots of carbons running around. And what does a kinase do? Phosphorylates, carboxy-enophyr. In this case, PEPCK. Everyone got that now? Phosphoenol pyruvate carboxy kinase. A little dash or space between the carboxy, yes. Biochemists in the old days loved these things. Fortunately now these days we go for lots of acronyms. So what's happening with the steroid, the glucocorticoid in your liver? It's enhancing PEPCK. What's it doing? It's taking pyruvate, making glycerol 3-phosphate. What does that do? Taking any fats, turning them into triacylglycerol, sending them into the circulation. What does it do in your fat cells? At that point in time, the same cortisol, which is probably happening in you right now, is inhibiting the gene that makes PEPCK in your fat cell. That means less enzyme to do the work. What's the enzyme doing the work? This glyceroneogenesis process, taking pyruvate to glycerol 3-phosphate. You are balancing out what's going on in your liver and your fat cell. Glucocorticoids like cortisol says if the liver, in this case, is making fat, what should your fat cell be doing? Really? If your liver's busy making fat, shouldn't your fat cell be accepting that and putting it to storage, and vice versa? Yes? Exactly, I know what you're thinking, yep. So this particular process, glucocorticoid put above that, cortisol, classic example, regulates level of PEPCK reciprocally in the liver and fat cells. That's why stress is kind of an issue. particularly chronic stress, like metabolism every week, labs every week. That is chronic stress. And what's that doing? Raising your cortisol levels. It's telling your liver here to get busy making more fat, isn't it? Telling your fat cells to accept it. But remember, that's dose dependent as well. Glucocorticoids can increase that value depending on how stressed you are. Now, what happens if you're acutely stressed? What's your liver going to do? Here's an adrenaline rush, an exam, metabolism right now. What's your liver going to do? Release glucose, isn't it? Get busy, run around, do nothing. So this process is a balancing act based on what's required at what particular time, but it's also talking about the chronic exposure to a glucocorticoid, aka cortisol. What does it do? It alters gene transcription. How much of that enzyme, PEPCK, is going to do those steps? Whatever is required, reciprocally regulated in different tissues. Can I move to the next one? Is that a yes? Before I do, just note this little bit of italics here. Remember I mentioned a couple of weeks ago that the number of fat cells you get when you're born are relatively constant? Yes, you might increase them and decrease them based on your diet. It's only when you keep on eating that you cause them to divide, differentiate, and have more of them. You keep them pretty stable. Now, if your diet's relatively, I want you to jot down, relatively constant, you're not going into one of those eating competitions. You eat relatively constantly, hopefully a nice balanced diet for most of the time, then this process is relatively balanced. It's only when you're diseased, chronically or acutely stressed that you're going to alter this. And when's a student chronically stressed, apart from metabolism? Exam periods. Well, you build up to it, don't you? And you will get exam questions from me talking about stress, so you'll know how to answer it, won't you? Sorry? You forgot Wednesday, 4 o'clock for Parramatta. And Thursday, 10 o'clock for Hawkesbury, okay? Who cares about them? That's right. They're not even farm people. That's the scary part. Okay, let's move on. Now, we mentioned glycerol as an energy source. I told you about 3-carbon glycerol can be helped to make what? There's my 3-carbon glycerol. We have a phospholipase, A1, A2, that rips the lipids off. What do I need? A glycerol kinase. Why do I need a glycerol kinase? What's the phosphate doing? Activating it, isn't it? There's my 3-carbon molecule. I have to activate it, ready to do some work. So I need a kinase to put the phosphate on. What I've made? Glycerol, 1-glycerol 3-phosphate in that case. So the position for the phosphate goes on carbon 3. That's essentially a one-way error. I've used ATP in my liver to make glycerol active, ready to do work. I need a dehydrogenase. In comes our NAD+. You'll notice that's a reversible reaction. Why do you think it might be reversible? What does the liver also have to do? Because remember, this is going to go on the liver, isn't it? What does the liver also have to do if I'm in a fed state? Make fat. But I need an activated glycerol, don't I? Just stick all the long-chain fats on. So it depends if I'm the fed or fasted. What's one of those short answer questions going to be? Fed, fasted, and stress states. I run down here to this isomerase. What have I done? Oh, kind of mixed it up a little bit. That's ready for glycolysis. We'll see that in a couple of weeks. Actually, no, we see it next week, don't we? Start glycolysis next week. Well, the semester's running quick, isn't it? So we're about to run down. That's a very simple thing. That simple thing happens in a couple of days of fasting. A couple of students at Parramatta said they're going to fast for a couple of days to see how it feels. Are you brave enough to do that? Very few people die from it. But see your GP if you're worried. I'm sure you'll survive. Now you're just in the what state if you're going through glycolysis with this glycerol starting point? What do you need? Sorry? Glucose, indeed. Or your system is going to try and make something, i.e. ATP is energy. Now, if the liver says, okay, I've got all this material, I've got my carbons ripped off, there's my 3-carbon glycerol, now I go into a fasted state, what am I doing? Making glucose. Now I suddenly eat breakfast because I've been fasting all night sleeping. What am I doing with that glycerol? 3-carbon glycerol, what am I doing with it? Just had breakfast. Making fat again. That's what your liver's got to do based on whatever the hell you eat at that point in time. That's how quickly it has to change. But keep in mind, if you're stressed about metabolism all the time, it's going to alter how much gene activity you have in the liver and the fat cells, isn't it? Which means they're going to be potentially raised up. And think about that process. You're elevating activity. So if I'm more fasting, then I'm busier doing that work when I'm stressed. If I'm more fed, I'm busier doing that work. Does that kind of make sense? I'm more efficient because I have more proteins to do the work, more enzymes. Can I get to the next one? Okay. Oh boy. I'll let you write and we'll talk about this one. Now this is, while you're writing, just write a little asterisk, very important slide. As opposed to just important. It's like an exam, isn't it? Pens clicking, writing away. Ready to talk about this one? Yes? The cell over on the left is a fat cell. Keep in mind, it's a white fat cell, not a brown fat cell. See those after semester break. White fat cells have hardly any mitochondria, and you'll see why after the break. White fat cells might insulate you, but they don't generate heat energy. Brown fat cells generate heat energy. Here's your white fat cell, and on the cell surface you'll see up there those tram tracks, which is the lipid bilayer, the cytoplasmic membrane. That thing there, the receptor, an integral membrane protein. What's that receptor for? Which hormone? And when does glucagon come up in your bloodstream? When is it elevated? In between meals, when your blood sugar gets a bit low, and we'll see the concentrations in a couple of weeks' time. When it gets too low, insulin starts to drop off, glucagon picks up. Now keep in mind, your bloodstream has levels all the time. There's no zero insulin, no zero glucagon. It's just the ratio change. Insulin drops off as blood glucose drops, glucagon comes up. What does glucagon tell your liver to do? Your liver. If your blood sugar's low, what would your glucagon tell your liver to do? Produce more glucose. But here it is on an adipocyte, telling the adipocyte to do something as well. Now this is, and I want you to jot this down, a simple signal transduction cascade, and that's the terminology. Pharmaceutical companies use it all the time. We use it in advanced cell everywhere else. This is what they're looking for to manage your cascade. A signal transduction. Think about, just look at the image for a moment. The signal is glucagon. Glucagon acts via its receptor, and it's telling the cell to respond. The signal transduction is the transducing of that interaction with that receptor, which could be physical, change in conformation, phosphorylation. There's lots of different ways that can happen. One of them particularly is that receptor activates this thing, a GS protein. Now, we go into detail next semester. Sometimes you do in physiology. This GS protein is stuck in the membrane because it has a little lipid stuck in the membrane. There it is, my GS, my hand. Finger is a lipid stuck in the membrane. Receptor comes along and says, oh, change shape, and run along and find this character. This guy here, write down AC, capital AC. See that acronym a lot. Adenyl cyclase. Big protein, embedded in the membrane. That just got a kick in the guts and told to do something by GS, usually GS alpha. You might want to put a little alpha on there as well. It's a multi-subunit protein. Adenyl cyclase, when it's being activated, turns ATP into cyclic AMP. Cyclic AMP, I want you to write next to that a common second messenger. We see that a lot in cell signalling, and that's again the terminology, signalling of the cell. Taking signals from the outside, telling the cell to do something. This is a biochemical cascade. You'll notice the dashed line, there's a whole series of steps. And I'll take you through them slowly. We'll go from the shallow end to the deep end. Put your water wings on. Someone asked me today, what are water wings? You know what water wings are, right, don't you? Those little puffy blow-up things on your arms as a kid. It's activating this thing, pKa. What do you think pKa is? Protein kinase A. Make sure you write it down. Protein kinase A. That's a ubiquitous signal transduction molecule. You know what ubiquitous means? Common. A lot of cells use it. What is it doing? Well, it's taking this blue blob, look at it. This blue blob is a protein, it's a lipase. That blue blob is an enzyme, a lipase. We saw lipase before, didn't we? Phospholipase A, 1, 2, 3, whatever. That lipase is sensitive to the signals glucagon sends, hence hormone-sensitive lipase. If glucagon activates this signal transduction, it tells this hormone-sensitive lipase to get busy in your fat cells and go to this lipid droplet, this thing here, full of here, these characters. Here they are, stuck in the lipid droplet, sitting there, dawdling around. It embeds itself in this membrane. Just jot that down. That's a lipid bilayer, that thing around there. The membrane of the lipid droplet is essentially a big vesicle. And if you see a fat cell, I'll show you after the break, it's huge. Hardly any nucleus, no mitochondria, the thing's full of fat. Once it embeds in that particular layer, because you can see here, notice the phosphate, I've turned somebody on again, as I was known to do in my early years. You love that, don't you? Turned it on, activated it with a phosphate, got into the lipid bilayer and snuggled up to this character, the little purple guy, and phosphorylated and phosphorylated and phosphorylated. That little guy is perilipin. It's already in the membrane, but when it's phosphorylated, you take those off, it hooks up with this thing. My God, this is a complex pathway, isn't it? Have you released any fat yet? No. This is glucagon coming up because you're not eating much, you need more blood glucose. Yes, liver's busy. You're starting to activate fat release after long periods. This perilipin hooks up with this CGI. Now, that's not that computer graphics enhancement stuff. I want you to jot down CGI is basically comparative genomic index, because what you're looking at is genome activation and comparison. It's not a protein we've found because we know it. We've looked at the genome and say, well, this is what it's possibly going to do. And what it does do is it hooks up with perilipin. It again gets released from that and hooks up with this protein, which is stuck in the membrane, ATGL. Not the Australian Gaslight Company. What the hell's that protein? That's an enzyme, adipocyte triacyl glycerol lipase. Oh boy. Man, this is complex trying to get fats out of your fat cell, isn't it? It's taken this triacylglycerol because what you've just done is activated it with its mate coming up nice and cosy, CGI. This ATG has taken that triacylglycerol and clipped off a lipid. You've started the process of removing a finger and sending out of that lipid droplet into the circulation. That's what these matches look like, except with the blue heads. You end up with a diacylglycerol, and that comes down to this character, which is also activated, monoacylglycerol lipase. That's also stuck in the membrane of your fat cells. What do? Start clipping off the lipids, send the fats out. Here you are sending the individuals, write down individual fatty acids into the blood circulation. Who's grabbed them? HSA, if it's you, BSA, if it's a cow, albumin running through your veins, grabbing the fat as it comes out so it doesn't coalesce with somebody else, transport it at a very short distance, a bit like an Uber driver, from here to there, that's it, out you go, bit of money. To this character, who's that? Who's a myocyte? You're all whispering, I can't hear you. A muscle cell, myocyte. Lots of actin, myosin, contraction, that's what it does. That myocyte will grab the fat running through this thing here, an integral membrane protein, into your muscle cell, and we start beta oxidation. We're going to clip off 2 carbons at a time. Whereabouts are you going to do that in your muscle cell? In your mitochondria. What do you generate from that? Lots of ATP. Your muscle cell also produces CO2. CO2 goes into your blood vessel. What happens there? Capillaries. Red blood cell takes up the CO2 in your big toe, converts it to bicarb in your big toe, puts the bicarb back in your bloodstream in your big toe. Red blood cell goes back to your lung, picks up the bicarb in your lung, converts it back to CO2 in your lung, spits out the CO2. What do? Exhale CO2. So your red blood cells carry oxygen, but convert CO2 to bicarb and back to CO2, depending on the tissue. Kind of clear on that? So this is signal transduction. It's a nice little way into how signal transduction works. It gets a lot more complicated, I can assure you. That's glucagon. That's when you're in the fasted state. What about when you're in the fed state? Insulin's going up. What should you be doing to a fat cell if insulin's going up? Storing more fat. And you'll see insulin dephosphorylates pKa and inhibits mobilisation of fats, so you don't release them so much. But I want you to also write on this slide, these fats, as I said earlier, don't sit in that droplet forever. You are constantly refreshing them. Muscles don't use them, they come back into the lipid droplet, they go back to the liver, they're constantly circulating, a bit like you see the freeway and the motorway. There's traffic all the time. It's not until work's required you get busy making those fats available for work. Because if they sit there all the time, they'll oxidise. We comfortable with that concept? Okay, lots of exam questions on that. Maybe time for a 5-minute break. Start back at 5. Get your insulin levels up, or your glucagon levels, depending on what you want to do. Lots. Some people get it more as they get more fat deposits. We'll talk about that after the break. When you have so much fat it can't get into the cell, it starts accumulating outside. It's like a delivery driver getting to the warehouse. I dump stuff, I'll just leave it on the door, and then we start seeing foaming macrophages, and then we start seeing all sorts of diseases related to that. Yeah.
Unfortunately.
Okay. Are we ready? Chris, are you ready? Chris is ready. Now this long chain fat, or free fatty acid, FFA, this long chain fat, what do they want to do? Marry. That implies copulation and offspring, that kind of stuff. No, they want to coalesce, they want to cuddle up. They're hydrophobic. You've just released it into the blood circulation. Who grabbed it? Albumin, HSA. I've just put it into a cell via a particular membrane transporter, an integral membrane protein. Here it is in your cell now, in your muscle cell. If I put lots of them in your muscle cell, what do these characters want to do? Coalesce. What do your cells do? Well, they have to find proteins that keep that thing from coalescing in your cells until they can start processing it. Here's one example. This one's well characterised. It's from the rat. It's called a fatty acid binding protein. Steroids have binding proteins. I mentioned CBG, corticosteroid binding globulin. That's a protein. Its other name is transcordon. Anything that's hydrophobic, someone will grab it and look after it so it doesn't do any damage in cells. You can see here, hopefully you remember from FP&G, these long sheets running through here. Most of those are hydrophobic amino acids. Grabbing the fat, keeping it linear, but it's accessible. So I want you to think about it a bit like my hand and the fat sitting in there nicely, but available for enzymes to use, but not available to make coalescence with other fingers. Is that kind of clear? That's important. So you've got the same kind of thing. It protects your cells. You know what the internal milieu means, don't you? The internal environment, the cytoplasm, the nucleus as well, protects that from lipid droplet formation. Back here, that is a lipid droplet, but what's it in? A lipid bilayer, a vesicle. So the cell is protected, the cytoplasm is protected, even though it's tiny, it's protected. So this beta clam type structure allows you to keep that long chain fat in yellow separated from some other long chain fat, because once they coalesce, just like the detergent in the sink, you need that to separate them again. That's a problem, particularly ones without double bonds. They'll stick like glue. That's a problem for your cells. But you just want that little clam so I can put the fat in there and then grab it for oxidation. So you've got an FABP in your cells. This one happens to be in your gut cells. Why is that? What happened in your gut last week? The epithelial cells after a meal. They're all whispering again. I can't hear you. What happens after a meal? Meal? What did you do? Remember that? Absorb those, take those two in, put them back together, but while they're in that epithelial cell, these are loose, aren't they? I've got to grab them so they don't do anything like that, and I can put them back on here after I activate them. I activate them in that beta clam. Same with your muscle cells. Comfortable with this one? Can I move on? What are we going to do now? If I see CoA, what am I thinking? What should you be thinking? You're activating somebody. You're going to do something with it. Here's our CoA molecule. What do we need? We need an energy supply. In this case, it's ATP with our phosphate group on it. What does that RC acid function mean to you? What's the R stand for? You're whispering again. What's it stand for? I can hear you whispering. Speak up. It's a finger, isn't it? It's a long chain fat, mostly palmitate. Just write it down. 16 carbon, could be any, but it's mostly 16 carbon. 16 carbon, because there you see fatty acyl-CoA synthetase. Hang on a sec, didn't we synthesise last week? What are you doing here? You've activated it with phosphate. You're going to switch it around with this fatty acyl-CoA synthetase to make what? Circle that. What's that? What's this stuff down here? Long chain fat with a CoA on it. It's active, ready to do something with. Now the thing about it is we have to, this stage, break this thing up bit at a time. 2 carbons at a time. So we're going to produce that, but as you can see in this particular process, it required energy. So that every time that long chain fat, let's go back here, every time this long chain fat gets into a muscle cell, the muscle cell has to supply ATP to activate the fat, ready for beta oxidation, ready for that long chain fat to produce energy, which produces more than glucose. Now there are a number of different isozymes. You heard about isozymes last semester, I hope. What are they? Isozymes. I know you did it in FP&G. And most of you did FP&G last semester, didn't you? Oh, I can't remember that. Collective amnesia. Or is that selective amnesia? Different forms of the same enzyme. They might differ slightly, but they do essentially the same job, depending on the tissue. Depending on the length of the fat, short chain fats, what are those? 2, 10, 12 carbons long. Intermediates, what are those? Maybe 16 carbon. What are the long ones? 22, 24, some 36. What do? Even the membrane lipids, remember those with the phosphate on? Those can be recycled. Those in your lipid bilayers can be used to make energy. That's what happens to you over time. Everything is hopefully recycled. If it's not, I want you to jot down it's necrotic. Things like gangrene, necrotic tissue, means you're not recycling. It's not going through programmed cell death. What have you done here? You've activated the fat ready for beta oxidation. Just jot it down. Ready for beta oxidation. Whereabouts? Mostly muscle cells. Can I move to the next one? Is that a yes? All right, this little convoluted, spongy looking thing on the left is a mitochondria. What are you going to do in a mitochondria? You're going to do beta oxidation. The problem is, that's a long chain fat. You saw those characters had trouble getting into your gut cell, didn't they? Especially when they look like that. And to get into a mitochondrial matrix, where I keep on saying, look at this inner membrane here, this convoluted thing. That's going to have the ETC set and all sorts of things. Remember I said it was a different lipid composition? It's trying to keep protons out. It's really hard to get lipids in there. They don't just flow in because you feel like it. They have to be transported in. To get them transported in, they have to be activated. So first you activate the fat. That's what you see over here. You see that R function? What's that? One of these fingers again. Long chain carbon. Here it is, this long chain carbon. We just back here activated it with CoA, didn't we? Yes? Here we are here seeing it activated with CoA. Now I've got to get it through here, the outer membrane of my mitochondria, through this intermembrane space. Keep in mind, lots of protons hanging around here, cytochrome C, and into the matrix. Last week, didn't I talk about a CoA pool, one for the cytoplasm and one for the mitochondrial matrix? The two cannot move between. So I've got to use one to do one in the outside, do something and get it on the different side and change it over. That's what we're seeing here. We've activated this fat. Here it is with CoA. To get this long chain fat into here, into the matrix, I have to run through this thing. There it is there, that blue thing like we saw a week ago, an integral membrane protein stuck in the membrane of the cell. Happens to be an enzyme called carnitine acyl transferase 1, also known as CPT1. Many diseases related to CPT1, often muscle insufficiency, lethargy, because people can't move long chain fats into their mitochondria for muscles to work really efficiently. So there's my long-chain fat. The fat, just watch my laser for a moment. The fat is going through this carnitine. The carnitine grabs it, drags it through this protein, which is a transporter called acyl carnitine translocase. You'll notice it looks more like a pore. So I've got my carnitine fat coming through. What happened to the CoA? As soon as that carnitine grabs it, CoA is recycled for somebody else. Go and grab another fat in the cytoplasm. Put a circle around this CoA. It's stuck in the cytoplasm. It cannot move into the mitochondria. So the carnitines grab the activated fat. There it is. It's gone through the intermembrane space, through this pore. Here it is in the mitochondrial matrix, but I have to activate it for beta oxidation. What do I need? CoA again. In this case, mitochondrial matrix CoA, the different pool. What do you do here? Carnitine helps me. In comes CoA. What have I got? Activated fat. Does that make sense? What do I need to do? I need to recycle the carnitine. This thing is carnitine acetyltransferase 1. I said, okay, thanks very much. I've transferred your R group long chain fat. You can go back out to the outer membrane and grab another fat and bring that one in. And that's what I want you to write down for carnitine. Please underline carnitine. It's not creatinine or creatinine. We're going to see that in muscle after the break. That carries energy around as a short-term supply. Carnitine is the carrier of fat from the outer membrane to the matrix. It gets recycled. It keeps the CoA pool separated. You see its chemical structure I put in here. Now, I don't ask for too many chemical structures, but you can see it's pretty simple, isn't it? A few carbons hanging off it, fairly hydrophobic, grabs the long chain fat, drags it through. Have you started beta oxidation yet? No. All you've done is put a finger into the matrix, ready to use. If it's less than 10 or 12 carbons long, you don't need the carrier. Why? Because the carbon length of the fat is short enough to get through without any help. So which carbon length fat would you think you need most help with? 16, palmitate, exactly. Plus any others that you're using. This will be an exam question, guarantee it. There's a little bit in the green box that's kind of important because this thing is inhibited mild by malonyl-CoA. Where did we see that last week? If we have malonyl-CoA, lots of it, what does that tell your liver cell particularly? You're busy making fats, aren't you? If you're busy making fats coming out of the mitochondria, why would you send them back into the mitochondria? That's why it's called a futile cycle. So you want to stop that futile cycle. I make a fat and it comes back in, that's pointless. So I inhibit that process. So malonyl-CoA doesn't just work to activate production of triacylglycerols, fat synthesis, it inhibits the process of fat breakdown, particularly at moving fats into the mitochondria. Man, it's complicated with fats, isn't it? Who would have thought? And all you want to do is diet and keep your weight down. But you need fat. I keep saying that. It's an important component. Can I move to the next one? Okay. All right, let's start beta oxidation. Let's watch one of the few animations we have. Whereabouts are we with beta oxidation? Which part of your cell? Can't hear you. A matrix of the mitochondria. Note particularly in muscle cells, okay? Muscle cells want to use fat. Higher energy source than glucose. Here is a long chain fat. It happens to be palmitate. It's a very simple schema, animation, I should say. On the left-hand side, you see the schema. Now, what I have to get across to you, which happens every time in the exam, there's my long chain fat. I said beta oxidation. Which one's that? The second carbon, the beta carbon. I'm going to clip it off. That's what those little blue squiggly bits are. I'm going to clip off that beta carbon. Every time I do, I generate an acetyl. How many carbons is that? 2-carbon-CoA. But I want you to write down initially, every time I cleave that bond, I generate electrons. Where am I? In the mitochondrial matrix. Every time I cleave that bond, I also produce acetyl-CoA. Acetyl-CoA can enter the citric acid cycle, and every time I run around there, I generate more electrons because that is going to be cleaved as well. So fat gives you like 2 levels of energy production. First, cutting the fat, 2 carbons at a time, and then running it through the citric acid cycle. And that's why you'll see fat has more capability to produce energy than glucose. That's why you are mobile. Trees are not. They use mostly glucose. When are they mobile? When they're in the seed form, and they use lipids mostly, because the density is much lower, the energy production is much higher. That's why you can move around. Otherwise you'd be full of water and would be having trouble moving around. Each one of these is producing some key energy carriers. NADH we'll see in a while, FADH we're going to see today. Circle those electron-proton carriers in the mitochondrial matrix. Let's watch the little animation. Remember, this is mostly a muscle cell. And in a muscle cell, what do you need? Lots of ATP. What do? You activate the fat. You do it 2 carbons at a time. That's why it's called beta oxidation, the second carbon. Beta oxidation, every time you clip off one, you generate electrons and protons and an acetyl-CoA. That's another 2 carbons. Those 2 carbons can go into the citric acid cycle, which is more electrons and protons. And we'll compare this with glucose and glycolysis in the next couple of weeks. We keep clipping off, and you'll notice we started with how many carbons in this animation? What was the fat? Your fat? Palmitate. How many electrons and proton carriers? And ultimately, when we start running around the citric acid cycle, a lot more ATPs. Some from the citric acid cycle, some from beta oxidation. Now, there's kind of some issues here in terms of how you do the overall equation, which I'll come back to. But remember, a 16-carbon fat, beta oxidation, lots of NADH, FADH, more ATP, and again through the citric acid cycle. Oh, there's the electron transport chain. Here's a little taste for you. Let's go back to this guy. Here's a long chain fat that you've broken up, generating all these energy carriers. What are you doing with them? Well, in your mitochondria, in your muscle cell, eventually, this here is the inner membrane. Didn't we say that that is different in composition? Up there, there's going to be lots of protons. Now, this is a segue into the stuff just before semester break when we talk about the electron transport chain. These 4 proteins are critical. These 4 proteins are going to move electrons and control them and move them through each other to this last complex, and that little blue guy there, that little football-shaped guy, is cytochrome C. This is all that beautiful Harvard animation we saw. What do? Each one of these is going to pump protons into the intermembrane space. So think about that. Each of your mitochondria are little batteries, protons being pumped out by you cleaving fat. Are you cleaving fat right now? Not so much. Let's watch those steps, and this is fairly basic. Electrons moving through. Every time an electron moves through one of these complexes, complex 1, 2, and 3, and cytochrome C, they pump protons out. Where do the protons come from? NADH, FADH, anything else you cleave as well. This last one, complex 4, that's where you use oxygen, which is kind of weird because you only use oxygen in one protein. You don't need that protein to keep cells alive. But if you don't breathe oxygen, you don't survive, do you? Why is that? Because you're greedy. You need lots and lots of energy, particularly for your brain. Protons come back in. Remember that Harvard animation, the spinning protein, ATP synthase? Here it is here. In this case, protons come back in and force the formation of ATP. I'm going to come back to this in detail in a couple of weeks. You need to know beta oxidation starts cleaving 2 carbons at a time. The 2 carbons as an acetyl-CoA generate more electrons and protons. So down the left-hand side, we have beta oxidation cutting up your fat. On the right-hand side, part of the citric acid cycle. Didn't we see that in the first bit of the semester, that things can come in and go out of that, like some amino acids? And I've said to you on this right-hand side of the citric acid cycle, that's where you cleave carbons. You break them off. What do you generate? CO2. You run around here and there's lots of electrons. Those electrons are in the form of FADH and NADH. You're containing them in the matrix until you give them up to those proteins in the inner membrane. When you do, those electrons, so think of that, your mitochondria are little batteries moving electrons and pumping protons. You wouldn't be here if that didn't happen. It's amazing biochemistry. And that's what you see down here, the respiratory. I want you to write down ETC, electron transport chain. That's what we're going to focus on. ETC. What does that ETC do? You saw pumping protons out, that moved that water wheel, which is protons in. What does that do? Forms ATP. This is what does all the work. So we're dealing with these overall cycles, beta oxidation, the citric acid cycle to generate these electron-proton carriers. Here's the overall equation. Now, I want to focus on this for a moment. 16 carbon, write down palmitate, fatty acid, generates 7 NADHs, 7 FADH2s, 8 acetyl-CoAs. You see the 8 acetyl-CoAs in brackets, those go into the citric acid cycle. Citric acid cycle, we have 8 times 3 NADHs, 8 times 1 FADHs, and 8 times 1 GTPs. Why the 8? From the 8 acetyl-CoA, 16 carbons, 2 at a time, that gives us 8, doesn't it? So you've generated a lot more. Overall, you generate 31 NADHs and 15 FADH2s. I mean, who cares? That's a hell of a lot of electrons and protons. That's a lot of reactive oxygen species as well if you're not careful, which means you generate an awful lot of what we call proton motive force, protons trying to come back in the matrix. If I'm pumping them out, it's mostly net negative, highly charged, want to come back in relatively quickly. And they don't disappear into your cytoplasm because of the charge difference in the matrix. Right, mitochondria are busy, aren't they? We've only just started. Can I move to the next one? Make sure you know the stuff for the exam. I'll tell you, there are lots of questions in the exam on this. Our friend. Last question in the exam, number 60. Here is a chemical structure. Well, here's one. What's that there? Base. What's that? Sugar. What's that? Phosphate linker. There's my bridge group. And then this thing, a couple of rings. Now, if you're like me and like hyperspectral fluorescence analysis, those things can be fluorescent. NADH, FAD, NADPH is the few that fluoresce in your cells. But that is an electron carrier. You see them all up here. Electrons, protons come in. I've got them for a short while. This is one chemical structure you must know for question 60. Is that kind of clear? What is this thing? Well, flavin adenine dinucleotide. Where do you find it? Derived from vitamins. What do we say about obese individuals? They can survive for long periods as long as they get vitamins, minerals, and some water. And even they don't need that much water. So you can then manage this process by managing all these electrons and protons in this character. Again, base linked to a sugar, phosphate linkage group. NADH, FADH, ATP. What did we see earlier? CoA as well, wasn't it? Finish with this one? Yes? Yeah? Okay. Oh my goodness. Look at the title, what does it say? Didn't we just talk about that a minute ago? Nice simple animation, nice simple schematic. Well, now we're seeing a bit of detail. That's all it is. What are we seeing in the detail? This process up here is termed enzyme channeling. It's a production line. Enzyme 1, 2, 3 and 4 work to do the work sequentially. What do? They're taking your cell, your 16 carbon fat, activating it, getting it running through and forming acetyl-CoA. That's all it's doing. It's not that complex. If you look at that little schematic and notice it's going from a 16, removing 2 at a time, down to this molecule. Here comes our fat. We saw that before. Down the right-hand side, you're seeing the enzymes. Write down enzyme channeling. That's more efficient. What are you taking here? An activated fat. There is my CoA. Whereabouts am I? In the cell? The mitochondrial matrix. What do I need? Enzyme 1 is a dehydrogenase. What do? I grab FAD, I generate FADH2. Isn't that electron-proton carrier we just saw a moment ago? What have I produced here? These characters here ready to do some work. I need a hydratase. What do I need? I need some water. What do I generate down here? This ketone function next to my CoA. I need a dehydrogenase. What do I produce here? Lots of electrons and protons. I take another step with this thing, the thiolase. What have I produced down here? What's that thing there? Acetyl-CoA. This is beta oxidation. These are the electrons and protons we see back here. Is that kind of clear? All you're seeing is the enzymes involved in this step. What happens to those electrons and protons? They go into the electron transport chain. They pump protons out. Here they are doing all that work over here, also known as the respiratory chain. So over here, I want you to write down ETC, electron transport chain. 4 protein complexes mostly, and cytochrome C. Guess we can watch the little video on this one. Do you want to watch the video? Yes? Okay. Getting bored, I can tell. Why is enzyme channeling going on here, do you think? It's a muscle cell. Muscle cell is working. Doesn't it need energy quickly? Shouldn't you break up these carbon bonds quickly? Here's the steps. I'll let it run through by itself. That's the same thing you see over on the right. Except now you're seeing the long chain fat down here, however long it is. There's the hydratase, same thing over there. Incoming water, same thing over there. What have you done? You've activated this component on the CoA, generated that double bond O. What have you produced? NADH. Electron, proton off. Electron, proton off. Electron, proton carrier. Here you've got your acetyl-CoA. Where does that go? Citric acid cycle. More electrons, more protons. So on a weight basis, fat has the highest energy production possible, at least in your cells. And you just keep on repeating it. Small issue for us, let's stop it right there. What about that fat with a double bond in position 9? That would screw up this particular process, wouldn't it? And you have to make some modifications, which you have to know as well. Talk about that. Now, I won't ask you if you're comfortable with this one. I'll ask you if you're finished with this one. Because I know you're not comfortable with it, because you have to memorise some enzymes. Are there any key players in here, do you think? Any key regulators? Think about your muscle. There it is. I'm telling you to work. Work, work, work. I've stimulated the process of beta oxidation. Now the muscle's working. What should it do? Generate lots of energy. That's why we have this enzyme channeling. Do it quickly. Break them up quickly. Generate electrons and protons quickly. Now, if I get you to get up and run around this campus, which is kind of hilly, you'll get out to maybe building 21 if we go that way. What's the next building over just here? 11. You'll start puffing by the end of 11, wouldn't you? Why are you puffing at the end of 11 before you get to 21? Unless you're an athlete. Why is that? Going to the InterVarsity games. Why is that? Not there yet, unless you're really unfit. Sorry, mate? Because your muscles, complex 4 needs that oxygen. You're working away very quickly. You're grabbing whatever energy. What are you doing to your fat cells? Starting to stimulate them to release fat. Who grabbed it? Albumin. What did it do? Send it into your muscle cell. What did that muscle cell do? Start this process. Now, if I had enzyme here, enzyme over there, enzyme somewhere else, it's going to take forever to grab this fat, cleave it, do all the steps we've just seen a moment ago through enzyme 1, 2, 3 and 4. Takes too long. Unlike we've seen other pathways, key regulators, this one says, as once you start, you go for it. If you've got fat and you're working, you make energy quickly. Does that kind of make sense? Because your demand is high at that time. You have a buffer. What was that energy buffer in muscle cells that I mentioned? Sounds like carnitine. Creatinine, keep that in mind. That'll have phosphates on it, kind of at the ready. You'll use them until building 11, maybe the end of 21. After that, you'll start stimulating fat release. If I keep making you running around, what do? Burning fat, aren't you? Instead of that, you're sitting here looking at me, making fat. Makes you feel good, doesn't it? Especially when I keep teasing you. But you do that, everyone does it. We finish with this one. Yes? What about medium chain fats? It's called omega oxidation. You've heard about omega fats, haven't you? Somewhere along the line. Omega fats is basically a minor pathway. Tend to break those down in the ER. Which ER would you think would happen to do that? Smooth ER mostly, yep. In this case, we're talking about hydroxylation that occurs on the methyl carbon, which is the opposite end of the fatty acid. You use monooxygenase, an enzyme, oxygenate NADPase. In this case, you're breaking up that shorter chain fat or this medium chain fat a bit differently. You're not going into the mitochondria. Most dietary fatty acids are even number. We talked about ingesting those fats last week, particularly triacylglycerols. You can manage them. You have a little bit of trouble managing odd chain fats. Who does with those? Well, plants and ruminants. Why a ruminant? What's a ruminant? Cow, sheep, camel, goat, horse. Why can they manage them even though they're mammals and you can't? Sorry? Yes, and what's in those stomachs that's helping them? Same thing in your intestines. Not just that, but who? Microbes. So ruminants have a capacity, but they rely on somebody else, microbes, to deal with those odd chain fats. Now, I'm not going to go into detail of those here right now, unless you really need me to, for whatever reason. Most of the animal people want to know about that. But in your case, you're a monogastric, aren't you? Those monogastric bugs will want to use up what you've got. You've got to absorb all these things as quickly as you can. Ruminants don't. They have 4 stomachs commonly. It's a rumen. It's a fermentation tank. That's why they get things like bloat and the like. Now, some of those produce things like alcohol and aldehydes. What do ruminants often produce that's a problem for global warming? Methane. Boop, there it goes. Not a lot of methane up. That's an issue because it's coming from the rumen, not from the cow's own physiology and cells, but from the rumen, other microbes doing it. Can I move to the next one? Yes? Okay. The other place that things get a bit weird is in your seeds. Not your seeds, your sperm or eggs, the seeds you might eat. What's unusual about seeds? Sorry? Wouldn't call germination of seeds unusual, but they're mobile, aren't they? Aren't they? I mean, they're not walking around, but they can move around, right? Which means their density is a much lighter process, at least outcome, than the tree is. Why? Because that's carbohydrates. All that stuff you're sitting on when it's not plastic, if it's laminates, is carbohydrates, a different linkage group. And hydrogen bonding stick together. That's what IKEA does. They heat up it, change the timber, cool it down, different hydrogen bond. But it's still a sugar. Sugar likes water. That's a problem. It likes a lot of water. Fat doesn't need water. It hates water. But fat is less dense than sugar. Less dense because it doesn't need so much water to be mobile. For a seed, for example, starts life, and I want you to note, particularly in this country, which is a bit unusual in this country, a lot of plant seeds in this country don't just use fat, they use amino acids as an energy source initially. Why do you think that would be the case in this country particularly, compared to, say, Europe, Asia? So, indeed, lack of water. Producing them. So they're a bit more efficient in that sense, but they still often use fats, which means they're mobile, lots of energy. What do? Well, on the right-hand side, there in the kind of browny colour, I want you to put a little box around that and put plant seed. Now, we used to do a seed germination track in Metabolism, but the university got really stingy and wouldn't pay for all the seed germination process. We'd take sunflower seeds, germinate them, one, to see all the fat that sunflower has in them when you centrifuge them. Curiously, I had somebody at Parramatta on last week trying to spin and mix cells in a centrifuge. Don't mix in a centrifuge. But when you centrifuge, you spin from density, don't you? Fat is less dense, so it'll float, won't it? In this case, we were getting seeds to germinate and looking at the enzymes involved in the first few days. You've all seen a seed germinating in primary or high school, maybe? What's the first few things you normally see from a plant seed? Not a grass, a monocot. What's the first two things to pop out? Oh. Cotyledons, thank you. Cotyledons, even though they've got chloroplasts doing some work, they are busy using these things. Glyoxysomes, peroxisomes. What are they doing? They're using the carbon in the storage of that seed to generate electrons and protons to work to make things while they start making themselves until they produce enough energy from the light sources, from the photons. That's at the beginning of the seed germination phase. And while that's happening, you and seeds aren't too dissimilar. That's why I put this up here to show you that seeds and you have very common pathways in the development of that seed, at least until it gets busy photosynthesizing. They're using what is termed the glyoxylate cycle. They will transition across to chloroplasts and photon excitation, which is incredible biochemistry because photons bounce around in that system to generate electrons in the PS1, PS2 system. So you're not too dissimilar. And what do you put into your fry pan often to cook things? Sunflower oil. Yeah? Canola oil. Sometimes you use olive oil. That's seed oil. As an aside. But keep in mind the first few stages, you're not too dissimilar. So a mitochondria and a seed, in this case, glyoxysome, peroxisome, have similar functions. It's a shame we don't do the germination prac anymore. It's a lot of organisation, and the university's more of a cheapskate now than it's ever been. Can I move to the next one? Yes? It's this one. So down here on the right-hand side, put a little box and write plant. Okay? Plant is on the right-hand side. A plant seed, particularly a lipid-based plant seed like sunflower, like canola, which is tiny. Did you guys germinate seeds when you were in primary or high school? Sorry? Alpha. That's horse food. Okay. Try growing some sunflowers at home. Can I move to the next one now? Yes? Okay. I mentioned a moment ago So what happens if you have double bonds? How are you going to manage that? Again, let's watch the animation first, but what you see on the right-hand side is the still version of the same thing. Can you guess, without looking too hard, what's likely to happen? Let's cover that up. There is a long-chain fat, couple double bonds, an eicosanoid being recycled, or arachidonate, thromboxane being recycled. They all get recycled. Otherwise, you have necrotic tissue. What's going on here? Isn't that just beta oxidation you saw just a moment ago? Until we get to those double bonds, then we've got to flip stuff around. Watch the steps. And these animations will be up on views for you to play with after I finish all this stuff. Slow fat breakdown. Linolenic. CoA activated fat, same as we saw before. We go through each step, beta oxidation. We get to this double bond. We can't do the same step. We need some other process to go on. We have to do a cis-trans isomerase, so you need an isomerase enzyme. You remove the double bond, you keep on beta oxidation, you get to this character. What do we do next? We've got to get rid of that double bond. We need some hydrogens. We bring them in. Here's an OH function. The epimerase is going to flip them around and get rid of the OH and give us a hydrogen. We're about now to go into the rest of the beta oxidation process. So even a double bond is not going to stop this process. Even a double bond in those fats are going to be used, which is what you see on the still version down on the right-hand side. You need to know this for the exam because it's a comparison of saturated fats and unsaturated fats on how you manage them. Would you suspect that an unsaturated fat, that is with double bonds, take a little bit more work to utilise? Of course you would. Wouldn't that be logical? Each enzyme here, we see the isomerase working away. We're running through the next cycle. We're using some electrons to do that. So it's a cost. Double bonds are a cost to cells. So that's why they like your cells like palmitate. Efficient to make, relatively, efficient to use. Efficient also means expeditious, happens quickly. Less enzyme involvement. Also less utilisation of other energy carriers, which means you're not wasting things just trying to produce energy from that. Your input costs are less if you use a saturated fat. Does that make sense? Your input costs. We're done with this one. So yes? Yes? Okay. Let's try and summarise some of that stuff. Are we just about done with fats for the day? We're just about done with fats for the semester? Not quite. They'll come back and haunt us for a while. TAG stands for what? Triacylglycerol. Are degraded into fatty acids and glycerol moiety through the cytoplasm of adipocytes. Remember the droplet. Who activated that? Glucagon through a PKA, hormone-sensitive lipase, perilipin, CGI. Our ATGL stimulated release of a fatty acid out into the bloodstream. Who grabbed it? Albumin. Transported into a muscle cell. Who grabbed it in the muscle cell? That beta-clam protein. They are transported. Make sure you put transported by somebody, not by themselves. Fatty acids go through as triacylglycerol, as chylomicrons, VLDLs, HDLs, or as individual fatty acids hooked up to albumin. It's amazing how the stuff you do in the lab just keeps coming back, doesn't it? If it's greater than 12 carbons, which is most of the free fatty acids you deal with, they have to be transported through the acyl-carnitine transport system. What did you do? Activated the fat, transferred it to carnitine. He dragged it all the way into the mitochondrial matrix, gave it to acetyl-CoA again. He got recycled and grabbed another fat. When is that occurring? Through beta oxidation, when the cell says, I need energy. Which cell mostly is going to do that? Mostly. Who's going to do that? Arnie Schwarzenegger, till he got old. And then it all went to hell, didn't it? So did his marriage and everything else, but that's his problem. If it's less than 12 carbons, it doesn't have the fat, the hydrophobicity. It can be easily managed. And where does it occur? In the mitochondrial what? Write it down, matrix. Who's going to do that mostly? Muscle cells. Is your brain going to do that? No. Make sure you write that down now. Your brain does not do beta oxidation. It needs to remember it, but it doesn't do it. Your brain does glycolysis. It makes a few long-chain fats to help Schwann cells, but that's it. We're not there yet, ladies. Fatty acids, I need you to note, particularly give you 2 times the energy outcome than carbohydrates. What's the carbohydrate we mostly talk about? Glucose. On a weight basis, a molecular weight basis, 30% of your energy comes from fat, so it's no good giving it up completely, because remember your lipid bilayers, even though a lipid droplet actually has a lipid bilayer around it, doesn't it? So it's no good saying I shouldn't have any fat. You must have fat. Some of them being used to make other things like cholesterol, steroids, bile, for example. And a 70 kilogram person, 70 kilograms. Around 10 kilograms of triacylglycerol I need a week. So if I don't eat, I don't last very long. I know some of you like to see that happen, but... And 80% of our energy needs are for heart all the time working, grabbing fat. Keep that in mind. If I'm releasing fat, and you've all seen pictures of live hearts, haven't you? Yeah, pumping away, but isn't there a little bit of fat hanging around there as well? Yeah. And as you get older, a bit more hangs around as well, which is what we don't need. But also the liver. Have I said much about the liver lately? No, I said the muscle mostly. The liver will do it. And what does the liver do with it? It makes acetyl-CoA. What does it do with acetyl-CoA? Lecture's not over yet, obviously. What does it do with acetyl-CoA? Make ketone bodies. Why is it doing that? Because it can't make sugar. And what's it doing that? Because your brain says, I don't get enough glucose, I need ketone bodies. Make sure you write that down on this last slide. I think I'm going to get dizzy doing this. So the liver's making acetyl-CoA onto ketone bodies. When is that happening? When you're working really hard, you don't produce enough glucose, and the harder you work, often the less brain capacity you have. Starting to run out of neuronal activity. You see those people at the end of a marathon, particularly women and men who are not really fit. They start collapsing, can't make decisions, can they? They've gone beyond that capacity. I know I've been close to that many times. Done with this one? Oh, what do you know? Now you can go, ladies. And what beverage do you need next week? Don't bring in all the same one, bring in something different. Manufactured, synthetic, nutrition label. Are you ready for the password? I had to change it up because too many people trying to guess. It's not HSL. I just did a COC for one of my PhDs this morning down at Parramatta. All caps, COC, confirmation of candidature. Alex, one of my supervisors. COC. Can I suggest to you, before you get too far, read the question carefully. should I say plural, questions carefully. I'll remind you that you will select topic A in the lab this week. When those are filled up, I'll deal with the other characters. I'm glad to see some of you get fun out of this.